How's it going? What's that? Really wet. I think it's not a trick question. Yeah, although we didn't get that storm they said we were going to have. It wasn't much of a storm yesterday, so. Although I think they got quite a bit north of here, so. Yeah, how's the studying going? At this point? You're all starting to think about it, right? So hopefully it's going well. I have not written the exam yet, so maybe you guys can give me some ideas for the exam, huh? I've done this in the past where I would let students submit questions, and then I would pick 2 or 3 of the questions. Is that a good idea or not? It is? Well, there's a strategy here, because then what students tend to do is they tend to ask the hardest possible question that they can think of, so they're the only one that knows the answer. And so they build their own extra credit. Does that make sense? So you still like the idea? The psychology of examinations is interesting. So, there's a famous psychological test where the professor says that they will give an A to everybody so long as all they do is they put their name on their exam and turn it in. And if everybody does that, everybody gets an A. But if one person takes the exam, then everybody gets a 0, except the person that took the exam, of course. Anybody who takes the exam gets points. That's murder, right? Would you like to have that option? Yeah. Well, actually, the answer is yes, because all you have to do is fill out the exam, right? So you just prepare for the exam as if you were automatically doing it. Then there's a whole group of people that think that they're going to get an A by getting a 0. I would never do that. But I mean, I've known somebody who's actually done that. Yes. I know somebody, I'm not going to mention names, I know somebody who did that, and I thought, what a dumb idea. What a really dumb idea. I mean, what you want in an examination is you want to see what people know. So when you start playing psycho games with people in exams, I mean, people are already stressed with stuff, right? So, yeah. Okay, so what I'm going to do is answer your questions. And I have no presentation, so I do these so that these are all question-driven things. We have the room for 2 hours. I don't think we'll be here the full 2 hours, but I am videotaping it for all the people out in TV land. And I did arrange to have the building unlocked. It wasn't the intention that you'd be locked out. The security people apparently did not get by. The last of you who got in, was the building unlocked, or is it still just propped open? He was unlocking. Okay, good. So they were supposed to unlock it about a half an hour ago, so I didn't realize that they had not done that. Anyway, welcome. On a scale of anxiety, are you very anxious? Raise your hands if you're very anxious. Somewhat anxious? I won't ask about the not anxious because your friends will kill you if you do that. So, okay. All right. These exams usually cause anxiety, especially the first exam. The first exam tends to have the most calculations on it and tends to have the biggest disparity between people saying they have enough time and people saying they don't have enough time. So that's because some people do the calculations fairly quickly. Other people struggle with the calculations quite a bit. So, word to the wise, get as much done as you can before you get to the calculations. Simply because you don't want to be throwing away points that you might otherwise have gotten if you were futzing with the calculations. I try to write all the exams to have the same length, but this one does tend to be a little bit longer, as I said, for some people because they're working on calculations. I have given you a sample exam on the schedule sheet, so I encourage you to look at the sample exam. Your exam will have the same format, but obviously the questions will be different. Surprise, surprise. As I said, I haven't written the exam, so I haven't even thought about it. We're doing a new book this year, so new things that I'm emphasizing, other things I'm not emphasizing, and so forth, and that's just another reason not to waste your time on studying old exams, because I think that's just a dumb idea. All right, that said, I'm going to shut up and turn it over to you. What do you want to know? Is that a hand? No thinking. Okay. Yes, there's a hand.
I have a question about random coils.
Yes.
Are they considered secondary or tertiary?
So a random coil is considered secondary or tertiary structure. It's, again, one of these nomenclature things that it's really, you could make an argument for it being secondary structure because they don't extend very far. And you could make an argument that it's tertiary structure because it doesn't have any regular form and, in fact, bends into 3 dimensions. So you tell me. What do you think? Okay, tertiary. It's tertiary. Yeah, secondary versus super secondary. As I've told several of you, I'm sort of wishing I hadn't brought that term out because I think all it's done is it's confused people. And my aim in bringing it up was actually to try to simplify things for you. Let me pull up protein structure. And where are we at? So there we all agree is a secondary structure, right? Alpha helix is a secondary structure. It's a regular repeating structure that arises from interactions between amino acids relatively close. In the case of the alpha helix, the amino acids are only 4 apart. Every 4th amino acid has some interaction between the one preceding it and following it. We also recognize beta strands as shown on the right. And beta strands, of course, look like that. Question? Oh, yeah. Okay. It's rare somebody says your mouth's not big enough, Ahern, so I'll try to do that. How's that? So, in fact, I made the videos yesterday for the problem-solving videos, and I try when I do those to speak softly. And so I'm used to speaking softly, which is not my usual manner of speaking. I used to when I used to lecture in Milam Auditorium, I used to be able to lecture in there without the microphone. If the microphone went out, I could always fill the room. I don't like to do that, so that's why I use the microphone in the new one, but I can if I have to. Okay, so beta strands arise from interactions that are close to each other. And when we put more than one beta strand together, we create what people call a beta sheet. There are other forms called beta barrels and things like that. And I simply use the term supersecondary structure to describe interactions between individual strands that are close to each other. They're not as close as less than 10 amino acids away. So it seemed appropriate that I shouldn't call them secondary structure. But it also didn't seem that I should call them tertiary structure because they weren't just random parts or 3D parts of a protein folded together, but rather they were things that arrange themselves in a regular fashion. So there's a supersecondary structure. There's one of those strands is a secondary structure. These two together are part of a supersecondary structure. So supersecondary structure, just secondary structures arranged appropriately with each other. That's all I use the term to mean. Okay? And again, don't get too hung up on nomenclature. I want you to understand the idea. That's the most important thing. Yes, ma'am?
The beta strand is just one of those strands?
Beta strand is one of those strands. That's correct.
So then what interactions are we looking at in the second?
So it turns out these guys actually are, they're not shown on there, but they are interacting with itself to give that beta strand the structure that it has. So, right. Yeah. So the terminology, secondary structure, primary structure, and so forth, different people use in slightly different ways, and that's partly from an historical basis. Okay. So historically, you had primary structure, which everybody agreed on, which was the sequence of the amino acids. Secondary structure were those regular structures. So you could see a fold, you could see a helix, and so forth. And as we started learning more about protein structure, we saw in some cases how these things were arranged with respect to each other. And so all of a sudden now you had something that was in between a tertiary structure, which was that overall three-dimensional form, and these repeating structures like you see of a sheet or a barrel or something like that. Okay? And so then what do you call it? So I'm not going to be picky about the nomenclature here, folks. I think if you have the idea about what they are, I think that's the basic thing. Tertiary structure, of course, is clearly something that arises from interactions between amino acids that are not close in primary sequence. And quaternary structure arises from interactions between individual protein subunits. And so those are pretty straightforward things. Yeah?
You went over a lot of purification methods, like the dialysis and the chromatography and
Yeah.
electrophoresis. And I was just wondering, do we want like nameplates under each one of those, or how much do you want us to focus on
Okay.
each one of those purification processes?
So a question about how much detail of the purification processes. Well, the guideline I would give you is the same one I give for everything else, which is anything I talk about in class is fair game. So I try to go through the main points of what I talk about in any of the methods, and I want you to understand how the method works. I want you to understand how you might use the method, what it would help you to do, those kind of a thing. Do you have a question over here?
Yeah. Can you go over the relationship between hemoglobin and BPG in the lungs?
Okay. Were there other questions? Let me, before I do that, other questions on the purification methods that arise from what I just said? Yes?
Can you go over SDS?
SDS?
Yeah.
Okay. So SDS is an acronym that stands for sodium dodecyl sulfate. And we use sodium dodecyl sulfate as a tool for giving proteins a uniform negative charge. And it does two things. One is that it denatures the protein, which causes it to stop being a coil, a glob, and makes it more of a linear structure, so it's more like a rod. And the other thing that it does is, of course, it coats it so that on the outside, all those negative charges of all the sodium dodecyl sulfates are shown. So the reason for doing that is that the separation methods that I talked about, agarose gel electrophoresis and polyacrylamide gel electrophoresis, both rely on the fact that you have rod-shaped molecules of a uniform charge, and the charge is proportional to the length of the molecule. So if you have a protein that's a glob, you, of course, don't have a uniform charge. You don't have a charge that's uniform with respect to the length of the molecule, and you also don't have a rod. So all those things are important things that SDS is contributing when you are doing electrophoresis. Once you have those things, then proteins separate like DNA fragments do on the basis solely of their linear size, because they are linear by that point that you've treated them with that. Does that make sense? Okay. Yes? The mechanism, okay, so before I do that, let me answer his question on this, and then I'll come back and do that scenario, okay? All right, anything else on protein purification? Okay, so you want to know about 2,3-BPG with respect to hemoglobin. All right, so if I go, let me see, I always end up with a song, don't I? Where are we at? Probably about. Where's my 2,3-BPG? There it is. Okay, so 2,3-BPG, of course, is a molecule that I said was a byproduct of sugar metabolism, specifically that of glucose. And the more sugar metabolism you have going on, the more of this molecule that will be present. So this molecule provides, first of all, an indication of how rapidly metabolism is going on in a given cell. And second, it affects hemoglobin. So the way that it affects hemoglobin is that 2,3-BPG binds in the donut region of hemoglobin. So I drew the hemoglobin as a sort of a donut-like structure. And that donut hole of hemoglobin is a perfect fit for 2,3-BPG, at least adult hemoglobin is. So adult hemoglobin, of course, has the alpha-2, beta-2 structure. And when you put those together, you make an ideal binding site for 2,3-BPG. When hemoglobin encounters 2,3-BPG, it binds to it in the donut hole, and that causes the hemoglobin to change its configuration from the R state to the T state and literally lock it in the T state. So it's really important that 2,3-BPG get out of hemoglobin. before it gets back to the lungs, because if it makes it back to the lungs locked in the T state, it's less likely to bind oxygen. It's going to bind less oxygen, et cetera. And so I talked about how that smokers have higher levels of 2,3-BPG in their bloodstream. And because of that, they are much more likely to have their hemoglobin make it back to their lungs, bound to 2,3-BPG. So their hemoglobin is going to be in the T state. It's not going to bind as much oxygen. And so consequently, smokers have reduced oxygen carrying capacity and therefore get out of breath when they climb stairs or exercise. And that's really what happens with 2,3-BPG. Yes, question? No, it does not. So that's a very important point. 2,3-BPG does not bind the heme. Okay? So it's simply binding in that hole in the hemoglobin and causing the shape change from the R to the T state. The only things that bind at the heme that I talked about in class are oxygen and carbon monoxide. That's the only two things that bind the iron in that heme group. Nothing else binds there. Everything else binds at other places in the molecule. So a couple hands, yeah. Yeah. So I know this is always conceptually hard to understand, but when we talk about binding, it's not like something is just stuck there forever, but that binding actually involves some fluidity. And the fluidity is what I describe as coming on, coming off, et cetera. We could think of this as like breathing, right? So something that binds tightly is going to breathe less. It's going to hold it more closely. Something that doesn't bind so tightly is going to breathe more, okay? Now, I don't know off the top of my head, but 2,3-BPG is probably in the middle in terms of how strongly it binds to hemoglobin. You don't want it to bind too tight because, of course, you want to get it off there by the time it comes back to the lungs. So during that coming on, coming off process, something that comes off doesn't have to come back on, okay? And so if it comes off and something else grabs it, like I say, like a cell or an enzyme or something that grabs it, then it won't make it back to the hemoglobin, and the hemoglobin itself will be free of the 2,3-BPG. And so that's what's happening during the travel back to the lungs that hemoglobin is doing. Okay? The 2,3-BPG is coming off, something's grabbing it, and it's a very useful metabolite because there's still energy that's in it. It's a breakdown product of sugar, but it's not a complete breakdown product of sugar, so there's still more breaking down that can occur to give the energy that would come from the sugar. So it's a desirable molecule for a cell to take up. Okay? Yes? So that's a very good question. Will carbon monoxide have the same effect on fetal hemoglobin that it has on adult hemoglobin? How many say yes? How many say no? Sorry, guys, that's a yes. It's binding that iron. That iron is wide open. This is you're confusing carbon monoxide with 2,3-BPG. 2,3-BPG, okay, keep these straight. 2,3-BPG doesn't bind to fetal hemoglobin. Why? The donut hole is different, right? So it's slightly different, very slightly different, but it's different enough that the 2,3-BPG can't bind to it, all right? So carbon monoxide will, in fact, bind to fetal hemoglobin. So when we talk about reasons mom shouldn't smoke, there's all kinds of reasons mom shouldn't smoke. One of those, however, is not that you'll get 2,3-BPG in the fetal hemoglobin because the fetal hemoglobin won't even bind it at all. But carbon monoxide, yes. Mom's reduced oxygen carrying capacity, yes. Two great reasons for mom not to smoke, let alone the nicotine and all the other crap. Yes?
So, does 2,3-BPG cause the cells to lose oxygen, or is it cells that have already released their oxygen?
Okay. So the question is, does 2,3-BPG cause the hemoglobin to lose its oxygen? Okay, go ahead. Okay. Okay. So the question really, I think, if I paraphrase it here, is what causes hemoglobin to lose all of its oxygen?
Does it play a factor in that?
Okay. So so the question is, okay, then I'll finally get the question out. That's fine. Does 2,3-BPG play an important role in releasing that oxygen? And the answer is yes, it does. So as we saw in class, hemoglobin can lose some oxygen irrespective of everything else. If you go from a high concentration of oxygen to a low, some of the hemoglobin is going to give up oxygen. That has nothing to do with protons. It has nothing to do with carbon dioxide. It has nothing to do with 2,3-BPG. And you want that to be the case. Think about it. You have resting cells. You have a lot of resting cells in your body right now. And they need oxygen. They just don't need a ton of it. So you want the hemoglobin to be providing a basic level of oxygen that a resting cell is going to need. And so that's working just fine. What I'm talking about with respect to protons, carbon dioxide, and 2,3-BPG all are signals that this is not a resting cell, that this is a cell that is needing oxygen because it is respiring rapidly. And it's respiring rapidly. It means it's trying to make ATP for some reason. Easiest to envision is a muscle cell, because a muscle cell needs ATP to contract. And as a consequence, those muscle cells will be needing this energy more. Oxygen, when oxygen's present, I think I said in class, the cell produces 15 times more ATP than when it's absent. Okay? That's a big chunk. So you want to have that oxygen there. So a cell that's rapidly metabolizing, you want to have a signal to give it the oxygen. So 2,3-BPG plays a role, protons play a role, carbon dioxide all play a role in that. Okay? Yes?
I'm confused what the relationship between histidine is on the hemoglobin molecule.
Okay.
And when there's carbon monoxide, carbon dioxide, and protons.
Sure.
So I know they don't, like where do they bind?
Yeah.
They bind to the
Okay. All right. So keep in mind that the globins, of course, the globins are the protein part of hemoglobin. The globins have many histidines within them. We talk about one that's holding that iron. And the only thing that does is hold that iron. It doesn't do anything else. So the other histidines that are in the molecule play roles in binding protons, binding carbon dioxide. Okay? So when we talk about binding of histidines to that's where it's happening. And it's for that reason that we say that carbon dioxide does not compete for the oxygen binding site, because these are other histidines that are grabbing that carbon dioxide. Right? So no other histidines, that histidine does nothing with protons or carbon dioxide. The other histidines that are in the molecule, histidine, why histidine? Well, histidine, if you recall, had a pKa value, or maybe you don't recall, I'll tell you, has a pKa value that's relatively close to physiological pH. It's about 6 to 6.5, depending on the environment that it's in. Okay? So that means that proton is fairly or relatively easily put on or off at physiological pH. So it becomes much easier to put a proton there on or take it off than it is to do it with, say, a lysine. Lysine has a lysine's amine group has a pKa of about 12. So at pH 7, that sucker is not going to come off, right? But if you have something that has a pKa in the physiological range, you're going to have a much greater chance of putting it on or taking it off. And so that's why the histidine side chain is actually used for the carrying of the protons. The carbon dioxide can bind to the same histidines that protons bind to. In fact, it will displace the protons if it binds to it. What does it do when it displaces the protons? Well, it, of course, increases proton concentration again, which means now those protons are going to go bind to another histidine, to another hemoglobin, and favor release of even more oxygen. So these two guys are working together to get this oxygen released. Another thing I should point out, and I didn't point it out in class, but I will say it here, I've mentioned it to a few of you, and that is that carbon dioxide is not predominantly carried by hemoglobin. Some of it is, a small amount of it is, but most of carbon dioxide makes it back to the blood by being dissolved as bicarbonate ion in the blood. That's the primary way that it moves. Okay? So when it gets back to the lungs, proton gets put onto the bicarbonate is converted back to carbon dioxide and water, and you exhale the carbon dioxide. So that's also a route. I didn't say that in class. Does that answer your question?
Yes.
Okay. Other hemoglobin questions before I deal with Sonora's question? Yeah.
Earlier, 2,3-BPG isn't like 14
Okay. So the question then has to do, the reason I'm repeating this is so people back there can hear that. The back there being the camera people, the TV people. Those people out there on Mars. So the question really relates to the binding of 2,3-BPG with respect to the release of oxygen. Does it happen before, after, during, et cetera? Okay. You don't really need to concern yourselves with that, to be honest. All right. But all that the 2,3-BPG is doing is favoring T state. That's really all it's doing. So what does T state do? It favors release. So if you want to think about it as the 2,3-BPG binding first and favoring the release of more oxygen, fine. But it doesn't mean that you have to have a hemoglobin that had no oxygen released before it bound or any of that. That doesn't have to happen. Yeah.
I found that histidine, does it matter what histidine in the protein the iron's bound to? Does it affect?
So does it matter which histidine in the iron in the protein the iron is bound to? Yes, absolutely, because when you think about the 3D configuration, there's only one that's going to be positioned right to do that. So yeah, it'll always be the same histidine because of the three-dimensional nature of a protein. Yes. Okay, so T and R states, are they specific to hemoglobin or do we see it in other proteins? We see it in a lot of proteins, and the most common proteins we see it in are enzymes. So when I talk about enzyme regulation, not this week, but next week, you'll see T and R is a very important consideration for that. So T and R really relate to, in enzymes, T state corresponding to the less active form and the R state corresponding to the more active form of an enzyme. Yes?
I have a question on this lecture. For people that have anemia,
Uh-huh.
and when you go from the round blood cells to the more moon-shaped ones,
Sickle cell anemia?
yeah.
Uh-huh.
So that happens in low O2 situations for both people that have anemia and don't, or just people who are anemic?
No. So the question is, when do you see the sickle cell-shaped cells? Does it only happen in people who have sickle cell anemia, or does it happen in other people who have low oxygen concentrations? And the answer is it's only in sickle cell anemia. Because only in sickle cell anemia does the hemoglobin polymerize under low oxygen conditions. And it's the polymerization of the hemoglobin that actually gives the blood cell the sickle shape. You don't see that in people who don't have that. What's remarkable, and I think I've talked to a couple of you in my office about this, what's really remarkable is that one amino acid change can convert hemoglobin from being something that doesn't polymerize to being something that does polymerize. And that one amino acid change, there are several mutations that can be associated with sickle cell anemia, but the most common one is one that converts a glutamic acid at position, amino acid number 4, to a valine. You don't need to know this, but I'm telling you this. To a valine at position 4. And that one change is enough to allow polymerization to happen. And you think, well, how does that happen? And you start thinking, well, if you try to put two glutamic acids together, what happens? They're going to repel because they're going to be negatively charged at physiological pH. So if you replace it with a neutrally charged side chain, that's apparently enough to allow the polymerization to occur. So there's probably other things around it that would allow polymerization, but the glutamic acid prohibits that from happening. Yes, Nick? Yeah, that's a good excuse me. A good question. This question was, does the heterozygous advantage that I talked about with respect to sickle cell anemia apply to only the one mutation I just talked about, or to all mutations that make sickle cell anemia? I'm not an expert in this, Nick, but I would assume we would see some kind of a spectrum associated with that. That is, in some cases, some mutations might have more of an advantage than others with respect to protection, but I just don't know the answer to it. Yeah. So I didn't say how it bound. I said where. What's that? Yeah, where. So it binds in the donut hole. So the donut hole is right There. I'm sorry? No, the iron, you can see the green guys here. That's the hemes. That's where the iron is, right? So, no, the iron is not located in the center. Yes, ma'am.
So you talked about vitamin C as a cofactor. Are there any other cofactors that you want us to know the specifics of?
So I talked about vitamin C as a cofactor. Were there other cofactors I wanted you to know the specifics of? Well, I did mention some coenzyme cofactors in the last lecture. So like anything else, if I mention it in class, it's fair game. So vitamin C, of course, came up with respect to the hydroxylation of proline and is essential for that process. A deficiency of vitamin C causes real big problems with that. Causes other problems as well. I'm putting together the lectures for the materials for this coming week's lectures. And I'm going to talk a little bit about reactive oxygen species. And reactive oxygen species are sort of made less harmful by antioxidants like vitamin C. So I'll probably say a little bit about that there also. Any more hemoglobin? Yes?
So what's the primary role of the heme and iron in hemoglobin? Is it for the histidines or is it for bonding?
So the primary role of the heme and the iron in hemoglobin is for binding oxygen. That's what the iron does. So if you don't have heme, you don't have the ability to bind oxygen. So that heme is essential for that.
Right. And is it just iron 2+, or can it be other?
Nope, just it's only ferrous iron that will bind the oxygen. If you have ferric iron, you have a form of hemoglobin known as methemoglobin, and it will not bind oxygen. And it's pretty much a toast at that point. If you oxidize the iron in hemoglobin, as far as I know, it can't be reversed, at least not easily. Yes?
What type of iron plus?
Ferrous, iron plus 2. Yes?
So if you don't have enough iron in your diet, would it remove just one of the irons on the hemoglobin?
So if you don't have enough iron in your diet, would it what?
Because like when you go to get blood work and
Mhm.
you have your iron level be too low, so
Right.
in that situation is that like the iron like removed from the hemoglobin?
So when you have your blood taken, they're taking blood cells, and the blood cells, of course, are what contain the hemoglobin. So the reason that they're concerned about anemia and so forth when you're giving iron is that anemia refers to having a reduced number of red blood cells. So that could be for a variety of reasons. Low iron could be one. Could be the fact that your body makes less. It could be that you have other factors like sickle cell anemia that contribute to a low count. So I'm not sure if I'm answering your question, though. Maybe I did. Okay. Okay, other questions on hemoglobin? Anything else on hemoglobin? Are we totally clear on hemoglobin? Yes, question?
This is back to my original question.
Okay.
I'm looking over like the notes with the cofactors, and it's kind of like, I don't know, they look really broad, so I just wanted to know if you want us to like look into that more, or if you want us to just know like the main points, like go back and just.
Sure. Well, so she showed me a thing relating to the cofactors and so forth. I guess my guideline, Megan, would be to look at the video if you have questions, because what I say will always be the most important thing. I'm sorry?
I was just saying you didn't expect us to know.
If I haven't talked about it, that's true always. If I haven't talked about it, I'm not going to ask you about it on an exam. Yes?
Can you explain the differences in the different binding states for allosteric enzymes and non-allosteric enzymes?
Sure. Let me answer Sonora's question first, since I bypassed her to finish up that, and then I'll come back to your question, okay? And Sonora, I can't remember what your question was. Mechanism of double displacement. Okay. All right. So these are both enzyme questions, actually. So double displacement mechanisms are pretty cool mechanisms. There we go. There's one. So in double displacement reactions, what's happening is that the enzyme is doing two things. It's doing a different thing to one molecule than it's doing to another molecule. And that's not what we saw in the other kind of multiple substrate enzyme reactions. The other multiple substrate enzyme reactions had either random binding or ordered binding. But the same reaction, that is, there was only one reaction that's being catalyzed in each of those cases. In the case of the ping-pong mechanism, you have two things that are happening. So the enzyme exists in two different states. And you say, well, why does it exist in two different states? Well, one state is required to catalyze one reaction, and the other state is required to catalyze the other reaction. And so the reason the mechanism is called a ping-pong is because it relates to the two different mechanisms, ping being one, pong being the other. And the enzyme just goes back and forth between the two. Now, there are many examples of ping-pong mechanisms. And if you've looked at the problem sets that I gave you, I invented a third mechanism called rock, paper, scissors that, it's theoretically possible, I suppose, that it could happen. I am not aware of any rock, paper, scissors. But if you look at it, you see the same sort of idea, that there the enzyme exists in three different states, and you can see it bouncing among those states, each state corresponding to one particular reaction that it's catalyzing. So when I look at the ping-pong mechanism, I have something like I have here with a transaminase. And in the transaminase case, you see the enzyme either carrying a nitrogen or carrying an oxygen. So if it's carrying a nitrogen, it's going to react with something that has an oxygen. If it's carrying an oxygen, it's going to react with something that has a nitrogen. So it's just back and forth between the two. It doesn't have to even be an atom. There's an enzyme called superoxide dismutase that carries an electron. The only difference is whether it has an electron or doesn't have an electron in the two different states that it exists. So a wide variety of ways in which they can work. So what we can see in this reaction is that the enzyme is catalyzing first the conversion of that alpha amino acid up above. All right. Well, actually, that's not even quite accurate from what I'm saying. Okay. We're seeing that the amine is going in the overall reaction from the alpha amino acid on the top to this alpha amino acid on the bottom. So what I'm showing you is the overall reaction, not the individual steps. The overall reaction is also moving that oxygen from the upper right down to the lower left. So the oxygen is swapping places as well. Well, this takes two reactions in order for this to occur. And the two reactions I summarized here, where I listed the enzyme first carrying nitrogen and the molecule A carrying an oxygen. So in the first reaction, the enzyme is swapping the nitrogen with the molecule A. And when that happens, it converts it into molecule B. Right? So I made the unfortunate decision to call that molecule B. I probably should have called it C, because we like to think of A plus B goes to C plus D. Right? But in this case, I've got A plus C goes to B plus D. All right? So it's a little confusing. But suffice it to say that all that really matters here is that the oxygen and nitrogen are swapping places. At the end, you end up with an enzyme that has an oxygen. The enzyme with the oxygen, the only thing it can react with is something that has a nitrogen. So that's the next reaction that occurs here. And then the enzyme's back to its original state. So it's just back and forth. This enzyme with the nitrogen now will go react with another one that has another molecule that has an oxygen. Yes? Are B and C the same? No. No. That's why I use different letters to distinguish them.
Okay, so A is reacting with an enzyme.
Yep. So if we were to write this reaction out, literally, like I said, it should be written as A plus C goes to B plus D. So I use different letters to distinguish the fact that they're different molecules. So A is converted into B. So we see A becoming B, and C is converted into D. That's what's happening. Yes, ma'am.
So I think why it's confusing
Uh-huh.
is because, like, if you wanted to draw it that way, that's fine. But if you're going to draw it this way, it should be like A is bonded to O, and then when it's in the B form, like A bonded to NH2 equals B, instead of it being B bonded to NH2. No.
Well, that is it. I mean, the only difference is that,
But I think that's why.
right? But I don't want to call that A-NH2 because, okay, here's a good example. So if you start with pyruvate, pyruvate is a molecule produced in glycolysis. It has an oxygen. If you compare that to the amino acid alanine, It's the same structure. You've just swapped a nitrogen and oxygen. So I don't want to call this pyruvate with a nitrogen. In other words, I want to call it what it is. It's a different molecule. It's alanine. So that's why I give it different letters. This is pyruvate. This is alanine. And the only difference is this has an oxygen. This has a nitrogen. But now this is a different molecule because it has a nitrogen. Right? So that's important to keep straight. I understand it's confusing, though.
So, in the second line of that equation,
Yeah.
first line you have A turning to B,
Uh-huh.
and the enzyme goes from having a nitrogen to having an oxygen.
Yep.
In the second line, you have a brand new molecule, C, that has nitrogen, but you have that same enzyme with oxygen.
Yep.
And then you're converting it to D and putting the enzyme back to the original state of the nitrogen.
That's right.
Okay.
Yeah. Did you start to say something? I cut you. Okay. Question? So is the ping pong mechanism and double displacement mechanism the same thing? The answer is yes. They are. They're both the same, different terms for the same thing. Double displacement. I like ping pong better, though. I want somebody to find a rock, paper, scissors. I think that'd be cool. Yes?
Wait, so double displacement is not random. Ping pong and double displacement are not random. Or are they?
So, the good question I'd ask you, is this random or not random?
In my notes, I had double displacement under random and then ping pong under a different category. So is that not correct?
So these are double displacement reactions that are ping pong mechanism.
Okay.
Okay? So multiple substrates will have, actually before I say that, before I make sure I didn't use that term, but double displacement reactions should be enzymatic reactions, single substrate, multiple substrates, and then ping pong. So you have that. Those are all multiple substrate, and there's double displacement. So this is a subset of the multiple substrate reactions. But double displacement is the same thing as ping pong. Now, well, okay. So his question is are double displacement reactions random or ordered? So they're really, okay, it's a little hard to say, right? Because when we talk about random and ordered, we had them both binding to the enzyme at the same time. That's where we had to, they both had to be on the enzyme at the same time so the reaction could occur. Here we've got the reactions displaced by time. The first reaction occurs, the enzyme lets go of it, then the second one occurs, right? So there's not a first or a second. It just depends on what state the enzyme is in. If the enzyme started with an oxygen, it's going to bind with a nitrogen. But if the enzyme started with a nitrogen, it's going to bind with an oxygen. So you could say it's random. You could also say, well, it's got to go in this order, right? But it doesn't coincide with the terms that I used before, which is when you have both substrates present on the enzyme at the same time, and you never have that with this. So I wouldn't categorize it at all. Now that I've totally confused you. Yes, ma'am.
What's the question that you asked about fire and cyclones and
Oh, okay, let me answer her question before I get to that. But you had a question about enzymes also.
I wanted to make the question clear now. So you talked about competitivity with relation to hemoglobin.
Uh-huh.
And then separately you talked about how binding of an enzyme to this substrate changes the function. And then I was wondering, why is it that the allosteric binding graph is almost identical to the hemoglobin binding graph?
Okay. So, her question, have you seen the problem set videos that I put up? No. Okay. Her question had to do with why it is that the binding of the oxygen by hemoglobin, why those plots resemble the V versus S plots that we see for allosteric enzymes. I'm going to refer you to the video on that, since I think I do a pretty good job, and it's a little bit of a lengthy explanation. That I think I do a pretty good job of explaining that there. And then if you have questions, let me know. How about that? Question? Oh, okay. Okay, I thought you were asking something else here. Anything else on enzymes before I move forward? Completely clear on enzymes. Okay. right, and your question then was,
I don't know. If you could just like go
oh, Ramachandran plots. Well, so Ramachandran plots are, I think, difficult to get your head around. And her question to me was, well, what kind of question are you going to ask? Well, I'm not going to tell you what kind of question I'm going to ask, right? Because if I did, that would be the only thing anybody would learn. And so I want you to understand protein structure, and Ramachandran plots are one tool for understanding protein structure. Ramachandran plots tell us, first of all, that there are steric hindrance considerations for protein structure, right? So Ramachandran plots are theoretical predictions for the stability of a protein. And I talked to somebody in my office, I remember it was you the other day, or somebody I talked to in my office the other day about this, where I tried to describe a Ramachandran plot as a three-dimensional plot. Wasn't you? Somebody, okay. So let me take a minute and do that, and maybe that'll help you to understand Ramachandran plots a little bit better. I'm going to put the screen up here. Can I turn that off without turning it off? Don't have a way. Okay, maybe that'll do it. Nope, that doesn't do it. Up. So a Ramachandran plot plots the two things that you can see on a graph. So the two things that you can see are phi angles versus psi angles, right? And so those angles are rotational angles, and they're rotations around the single bonds of the alpha carbon. So we've got a double bond-like structure for the peptide bond. We've got an alpha carbon, and we've got an alpha carbon. So here's the alpha carbon, here's the alpha carbon, all right? This is peptide bond here. Now, I don't care if you know that this is a psi, or this is a phi, or this is a phi, or this is a psi. I don't care if you know which one is which. That doesn't matter to me. All right? You should definitely know it's not this one because this one doesn't rotate, right? But these guys can both rotate. And because they can rotate, that means that all this other stuff that's hanging off of them, remember we have an R group and we have an R group. All these other things that are hanging off of them now can bump into each other. So the question then is, well, where do they bump into each other and where do they not bump into each other? Right? Well, phi psi angles measure that. To rotate something all the way around a circle, of course, is 360 degrees. So we plot these from minus 180 to plus 180, which is 360 degrees. We could just as easily do it as 0 to 360. It doesn't really matter. But the convention is they've used minus 180 to 180. When they do this, they find certain angles are associated with stability. That is, that there's no problems with steric hindrance. And those areas are roughly looking something like that. It's a little bit bigger, okay? Where this corresponds to an alpha helix, this corresponds to an alpha helix, and this corresponds to beta strands. And you say, what does that mean? Well, how did you get this information? And so the way that this information comes about is by taking every single angle and saying how close are all of the groups that are out there. And we know from van der Waals interactions, we know from how close we try to put nuclei together, whether that is stable or unstable. The tighter you try to put together two nuclei, the more energy it takes to do that. Okay? The energy it takes to put them together goes up to the power of 12 when you get to certain short distances. It's enormous. Okay? So the forces that will resist steric hindrance are strong. And they're a function of distance. So you can tell a computer program, well, look at all these angles. How far is everything apart? Here's the distance between this atom and this atom. What's the force, okay, that's associated with putting it there or taking it apart? Okay? So for each one of these, we could imagine that there are some places, like let's say right here, where that force is impossible. We're not going to beat that. It might take an energy that is really, really large. Something right here might not take so much energy. So if we think about this in 3 dimensions, we think something that's way out here, it's a lot of energy, and something that's way down here, close to the surface of the board, is very little energy, corresponding to the angles. And so these things arise then as the low points in that energy diagram. These are the lowest points. The least energy is where these things are going to be stable. Does that help? Okay. So, way out here, not stable, and that's why we don't see anything popping up. But these guys right here are the lowest points, and those lowest points of energy turn out not to just be low energy points, but they turn out to be related to actual structures. And so nature obeys the theory that we associated with that.
It would be a lot easier if they could somehow model it more like a 3D, like you're saying, instead of.
Yeah, you know, I had never, I can't remember who it was I was talking to. I was talking to somebody in my office about this, and I had this idea of describing it in 3D, and I thought, you know, it does make sense that people should describe it that way, because now you understand what this really is. Right. Yep. But now everybody's going to make an A on that part, right? Okay. Let me get back to laptop. Okay, other questions? Yes, ma'am. So the forces arise from steric hindrance. That's right. Yep. And you can imagine it's pretty complicated there because you've got all these things hanging off, but then you've also got things further down that can get in the way. If 2 out of every 3 bonds can rotate, you got some enormous number of considerations of things that can bump into it. So the reason that the cis configuration of the R groups is unfavored is because of steric hindrance. That's correct. Yes. Question back in the back?
Yeah, for the parallel Vegas string,
Yes, sir.
you have the R groups drawn right next to each other.
Mm-hmm.
So that would be when you get
You're referring to the figure I had up on the board about before? Okay, let me pull it up and we'll make sure we're talking about the same thing. That would have been protein structure. And this figure? Okay. Now, what was your question?
The R groups are right next to each other, so there'd be high steric hindrance there.
The R groups are, oh, you're talking about like right here in the middle? Yeah. So that could be a factor, certainly. So one of the things that you see is that there are certain amino acids that are favored in beta strands and certain amino acids that aren't. I think I showed a table of that. Okay, so that would be reflecting what you're talking about here. You see the same thing with alpha helices, but it doesn't even have to be with between. It can also be within. Quick question, yeah. Mm-hmm. Yes. I believe I fixed it. Let's see. Yeah, they're fixed now. They're fixed. Yeah. I went home that night and fixed it. Yeah. Yep. Now you'll be able to draw that on the exam, right? I didn't say either one was favored. Okay, they both conform. Yes? The proteins that what? Oh, inherently disordered proteins, yes. Okay. No, in fact, we've got a person coming in January, I believe, who's going to give a seminar on inherently disordered proteins that I'll announce to the class when he comes here. And his contention is that they're way more widespread than people think they are. So they're not oddities or rarities, no.
So just to clarify one, like in terms of stability, you said neither or
I said I didn't say one versus the other was. Yeah, I wouldn't say neither is, but I didn't say one versus the other was. Would you like me to make a determination so that way you have something else to memorize? I think you don't. Okay. Yes, Nick.
Can you talk about the hydration
Hydration
of the peptide?
meaning?
Like the formation of the peptide bond.
Oh, okay, so the dehydration or the hydration would be involved the breaking of the peptide bond. Okay, sure. So that actually goes back to here. And so, two amino acids brought into close proximity. We see the alpha carboxyl of one, the alpha amine of the other, that combine. Water gets split out, which is what's happening right here, and the peptide bond results. The alpha carbon, of course, is here, so you've got phi and psi angles associated with these two, right? And you'd have phi and psi angles associated with these two as well. Okay? Yes, ma'am.
you talked about double bond formation. So that happens when the carboxyl oxygen becomes a single bond, right?
So resonance gives rise to the double bond-like character of the double bond, of the peptide bond, and that arises as a result of migration of electrons from here into here. Yes. Yes. So resonance is a something that you hopefully learn in organic chemistry. It's not a stable, continuous structure, but it's possible. Resonance referring to multiple things that it can be, switching back and forth. So you think of your voice being resonant with something. Mine isn't resonant with anything very good, but it makes it is a fluid thing. Yes, sir.
Having trouble distinguishing between a couple of the characterizations and purification methods.
Okay.
What's the difference between centrifuging cells and fractionation?
So what's the difference between centrifuging cells and fractionation? So fractionation simply refers to a process of separating things.
Isn't centrifuging a type of fractionation then?
Yes, it is. So centrifugation is a type of fractionation. Anything that causes two things to be treated differently in terms of isolation is a fractionation. Yeah.
Okay. And then with polyacrylide gel electrophoresis,
Uh-huh.
how is that different than agarose gel electrophoresis
Okay.
other than the use of SDSP?
Okay. So the question was, how does agarose gel electrophoresis differ from polyacrylamide gel electrophoresis other than the SDS part that I talked about with proteins? Yeah, that's a good question. The only real difference has to do with the size of the pores. So when you use agarose gel, you have quite relatively large pores, and it's the pores through which the molecules are being driven by the electrical current. So the pores provide the separation means, and so the pores in agarose gel electrophoresis are gigantic compared to what they are in polyacrylamide gel electrophoresis. So that's for that reason that we use agarose for DNA. We almost never separate proteins by agarose gel electrophoresis because there wouldn't be any separation. The pores are so big that the protein just goes shooting right through them.
Those would be for like nucleotides.
For DNA or RNA, not nucleotides, because nucleotides are the monomers, right?
Okay.
Yeah.
So like polyelectrophoresis is for RNA?
So polyacrylamide electrophoresis is, we separate proteins only by polyacrylamide electrophoresis, polyacrylamide gel electrophoresis. We can also separate small DNAs and RNAs on polyacrylamide gel electrophoresis as well. But most DNAs we separate by agarose. Yeah. So the size difference of these is enormous. DNA is way bigger than any protein, way bigger. Yes, ma'am? The what? Yes? That's the resonant thing I was talking about a second ago. That's right. So a peptide bond behaves as if it were a double bond because of resonance. But it's not a double bond. But it behaves as if it is, and we can treat it as if it is for that reason. So that's why we think about the where I showed that here's this hand was a plane of a peptide bond. Double bonded things will be flat. You learned that in organic chemistry, right? So it's flat. So this is the peptide bond. On the ends are the bonds where my finger and thumb are the single bonds that connect to the alpha carbons, right? So the alpha carbon, if the alpha carbon is my nose, then here's the next single bond over here. And so the rotation that I'm talking about is this, right? And we see that the peptide bond is contained within that plane. Does that help? It is not drawn as a double bond, no. No. Yes, ma'am?
So proteins have some kind of structure. Is it that having that partial double bond structure allow it to have some like baseline like structural integrity, and also the rotations of the ion side give them the flexibility?
So I think what you're saying is, does the peptide bond, because it has double bonded character, provide a more stable structure compared to the flexible structures of the single bonds? Is that stating your question correctly? Well, proteins have a stable structure not just because of the peptide bond, no. I mean, those bonds, these single bonds are stable, okay? They're just flexible, right? Or rotationally flexible. But I mean, they're not flexible in the sense that they're doing this, okay? They're just simply rotational flexing that's happening. So the stability of a protein is not just due to the peptide bond. No, I would say that's wrong. Yeah. Yeah.
Broad question, historically, what students struggle on the most doing the test?
Historically, what have students struggled the most on this test? Well, since you're the first one taking it from this textbook, there's no history. I don't know. You know, if I said one thing versus another, then I think what that does is it cues you to work on something that might not even be a problem for you. So I would be reluctant to answer that question because different people struggle with different things. And I just am, I think that you should always approach the exam by how well do you know the material, how well do you understand that material. And so I think that's, I mean, I know it's kind of a cop-out answer, but I don't know any way to do that without, as I said, influencing you overly because of something I might say. I had a question over here first. Yes?
I'm having like a really hard time with chromatography, the cation and ion chromatography.
Anion?
Anion and in the textbook and the notes, especially with the molecule that goes from sodium to calcium. I can't remember if it's in the notes in the textbook or both.
Mm-hmm.
I don't really understand like what the purpose
Okay.
of changing the
Okay.
ions are.
All right, so let's take a look at that. That would be characterization. And it would be fairly early. You're talking about this figure right here, right?
Yeah, what's the? I kind of understand what you're doing with the because Na is only just plus and calcium is 2 plus, but I don't understand like why what it's telling us.
Okay. So I only put this figure on here to show you a way that you can bind, okay, positively charged things using a cation exchange resin. So any resin that you use will always have a counterion. In this case, the counterion that you start with is sodium. But if I put a protein on there instead of calcium, then the protein would bind and the sodium would come off. So the calcium just what I happen to have in terms of what I was going to bind to this resin, right? But the principle is that the counterion gets washed away, the thing that I've added to it binds, and it sticks, if it's the opposite charge. And it is. Right?
Right.
So this could be a protein. So if I have a positively charged protein, all right, then right here, I'm going to have a protein stuck. Make sense?
Yeah. So what are you trying to identify in this type of?
I'm not trying to identify. I'm trying to separate. This is the fractionation thing. So I might be trying to separate something positively charged from something negatively charged. So if I added, let's say, a bunch of negatively charged protein to this, it's not going to stick because it is going to be repelled by the resin, right?
By the calcium.
No, by the resin. The resin is negatively charged,
By the
right? But a positively charged protein would be retained,
Okay.
right? So the ion exchange resins allow you to separate on the basis of charge.
Got it.
Okay. Question?
I was just going to ask in terms of the calculation to separate from log times sticking to 12 time test,
Mhm.
where would you go besides the study questions to get the repetition of
Where would I go besides study questions to get repetition for calculations questions? There's an internet out there. That's what I would advise. Yeah.
Just the test in general, should we be prepared to draw structures?
Okay, so the only structures on an exam I will ask you to draw will be structures that I told you you would have to draw. So since I've told you you don't have to draw any structures so far, there's no structures there. But that doesn't mean you shouldn't know something about structure. You should know, as I said, how each amino acid ionizes if it does. Right? You should know the categories of the amino acids. But that doesn't mean I'm going to say draw alanine. Right? Yes, ma'am.
I have a question about the different charge for protein given the pH. So if it is within the plus or minus 1 margin of pKa, do we assume it's neutral?
So so the question had to do with the charge of weak acids with respect to pH and pKa. So I said that if the pH of a solution is more than 1 or more units above the pKa, the proton is off. If the pH of the solution is 1 or more units below the pKa, the proton is on. And then her question is, well, what happens if it's within that? And the answer is, well, how would you know? What would you know? You wouldn't know anything. You'd know that some of the molecules have the proton on and some of the molecules have the proton off, right? Well, it would be dumb for me to ask a question saying which one it is. No, I wouldn't say it was net neutral unless I had it exactly at the pKa, right? And I always want to be careful with the word neutral. Neutral to me means 0, right? But neutral might mean plus, neutral might mean minus, right? So I don't know what neutral means. Okay. Yes, ma'am.
For the enzyme catalyzing, it's being quicker than a chemical catalyst. Is it because it has specificity?
Okay, so you're asking the question from the problem set? Yeah, I posted videos last night on problem set 6 and 7. So I'll let you look at that. So I think that's answered pretty well in there. Yeah. Yeah, the reason that it takes so long to get the problem sets done and everything is I'm making them up as I go, folks. So because they weren't written for the book when the book was done. So I'm writing the problem sets as they go, which means I have to go do the videos as I'm going. So I did the videos yesterday for problem set 6 and 7. So my day was kind of shot yesterday. Did anybody go to the football game yesterday? It wasn't wet, was it?
I have a question that has to do with the ionization.
Yes.
For the worksheet, it might be easier for you to see when the pKa is see like above the pH. Is there like a rule that says, oh, that proton gets pulled off to the remaining group first, or that proton gets pulled off to the carboxyl group
Okay. So our question really has to do with how ionization happens within an amino acid. So you've got an amino acid, for example, that might have an alpha carboxyl, an alpha amine, and an R group carboxyl as an example. And our question is, how do you know which group comes off first? Somebody else want to tackle that instead of me? It's the pKa. So the pKa is the thing that determines where the proton comes off first, because if you remember, a low pKa means a stronger acid, a stronger weak acid, which means more likely that it's going to ionize before something with a higher pKa. So the pKa of the alpha carboxyl, for example, is 2.2. So that is going to ionize before the R group carboxyl, which has a pKa typically of about 4.4.
Oh, the inverse. Okay, yeah.
Right?
Yeah.
Yes, ma'am.
What is a sphingolipid?
What is a sphingolipid? Have I said sphingolipid in class? Where did I say? It's in my highlights. Oh, I was probably talking at the very beginning about amphiphilic molecules. Is that? Okay. I apologize for that. Sometimes the biochemistry is so terminology laden that it's hard to make sure that the first time you talk about it, you define it. A sphingolipid is a, you don't need to know this, by the way, but a sphingolipid is a molecule we'll talk about later, commonly found in membranes. And it has characteristics similar to that of glycerophospholipids, or phosphoglycerides, as you probably call them. So it has a polar part, it has a nonpolar part, and I would have talked about it relative to amphiphilic molecules. So sorry, yeah, put panic into you. Yes?
Could you explain
Amphiphilic molecules, okay. So I talked about molecules that were hydrophobic and hydrophilic. So a molecule that's hydrophilic is something that associates very well with water, dissolves in water. Acetic acid, for example, is hydrophilic. It makes hydrogen bonds with water very readily, dissolves completely in water. Okay. Cholesterol, on the other hand, is very hydrophobic. Fat is very hydrophobic. It doesn't form hydrogen bonds with water, and consequently fat or oils will separate from water because they have no way of associating with each other at the molecular level. All right? In between those two are molecules that we call amphiphilic. And amphiphilic molecules are those that have characteristics of both being hydrophilic and of being hydrophobic. So the example I gave of an amphiphilic molecule in class, I think, was a fatty acid, which has a part of it that has a carboxyl that can readily form hydrogen bonds with water, but then it has a long tail that doesn't like to associate with water at all. So amphiphilic molecules will usually have a sort of an odd solubility in water. Soap is made up of fatty acids, for example. And so soap makes micelles where the long polar tails associate with each other away from water, and the outside part is where the carboxyl groups all associate with water. And it's for that reason that soaps, for example, are useful for dissolving grease in water. That's why you wash your hands with soap, because that micelle can actually take little fat or little oil globules, grease globules, and so forth, and sort of hide them away from water. Yes, ma'am? Is what? Is sodium dodecyl sulfate amphiphilic? It is, yes. Any detergent is amphiphilic. Yes? When you're reading a gel, what causes some, you mean? Uh-huh. I'm not sure. I'll pull up a gel, and maybe you can point out what you're talking about. Let's see. There's a gel. This what you're talking about? Yeah. Okay. You're talking about the smear? Yeah. Well, it depends on what you've been doing. I mean, sometimes people don't load things well, and so you may get smears associated with that. You may have been working with, let's say, a mixture of a large number of DNAs that have gotten broken, and so because they're very widely varying sizes, each size will come at its own. You won't see a distinct band, but you'll see a sort of a continuum of sizes with that. And third, sometimes remnants from previous gels. People reuse their gels. So there's a variety of reasons that that could happen. One of the things I learned when I first started doing serious molecular biology was the importance of having very, very clean materials for running gels. The methods that we were using at that time, they're radioactive, and the methods were very, very sensitive. So if you're trying to detect something that, let's say you wanted to detect bands within a gel that you couldn't see with your eye with a dye, but that were present, the radioactivity would tell you that. But if somebody hadn't cleaned out the gel apparatus before you used your gel, or you didn't clean it out before you used it, so you had the gel apparatus had all these other molecules that were in there, some of which might be the thing that you're detecting, what would happen is you would get just this giant thing that it would be black because you'd have all the contaminating things in it that you couldn't see with your eye, but that were definitely there. So, yeah. Yes?
I think at one point you mentioned that intrinsically disordered proteins are a type of protein regulation. Could you explain that?
So intrinsically disordered proteins, I didn't say were a type of disorder, were a type of regulation, but that their disorder might play a role in their regulation.
Okay, what is that?
Okay. So imagine that I have, so do you know what I mean by when I say an intrinsically disordered protein? Okay. Well, I draw you structures of proteins on the screen, and you can see, okay, here's this bend, and here's an alpha helix, and here's a random coil, and this is the way that hemoglobin looks, for example. That's the way it looks. And hemoglobin has a relatively fixed structure. A little bit of flexibility, right? That's how we get R and T states, but really it's pretty much, structure is pretty well set there. There are proteins that don't have such a fixed structure. At any given time, they can be all over the map. In fact, if you look in the book, I have, I think, an intrinsically disordered protein. You can see all the various wiggles and jiggles that it has in it. Well, if you have a protein who is functional only when it has a specific structure, then things that put it into that structure will activate it or inactivate it, depending upon the nature of the protein. Some proteins interact with each other. And when they interact with each other, they start out mixed up, but the only way they can interact is when they lock into state, right? So once it locks into state, it is bound to this protein, or once it is bound to this protein, it has locked into state, in which case, once it gets locked into state, it is going to be active or inactive according to that. So that's what I'm talking about with respect to regulation. Yes, ma'am?
A long time ago, I might have misconstrued what I was told, but prions were from animals, like cows eating other cows, and that idea, and you said you don't really know why they form.
Yeah.
Is that
So,
theory?
yeah, good question. It's a good point. Prions, of course, are the infectious protein particles that are associated with things like mad cow disease. And the question was, how do they spread? And one of the ways in which they're believed to spread is by organisms cannibalizing other organisms. So if you're a human being and you are feeding cattle byproducts of cow, then the concern is that some of those byproducts of cow might be containing prions that the cow could pick up and get. So you're exactly right. This incident that I talked about in England back in the 90s arose because they were not screening what they were feeding to their cattle, and it turns out they were feeding cattle byproducts of cow. So after that, mad cow disease went way up. Pretty good evidence that there was something associated with that. And when they stopped doing it, the incidence went way down. So you'd say, well, there's the cause. Well, yes and no. We don't feed that to cows, all right, and we still have mad cow disease that arises. We have people who don't eat other people, but who get the equivalent disease called Creutzfeldt-Jakob syndrome, okay? So, yes, eating another organism that already has that disease can certainly contribute to the likelihood that you'll get it, but it's probably not the only thing. So we don't fully understand how that happens. I had a friend whose grandfather or uncle or something was a coroner, and came down unexpectedly with Creutzfeldt-Jakob syndrome. And they felt that he had probably gotten it by close contact with a corpse that had had the syndrome. So it's probably might be a hazard of the job. I just don't know. Yeah. So yes. So aren't proteins supposed to be digested in your stomach? People argue about whether or not that happens with the prions. The argument against that for a prion was that after the incidence of mad cow disease arose in England, a variant of Creutzfeldt-Jakob syndrome in people arose almost coinciding with the consumption of what was unscreened beef in England at that time. So that would suggest that there's something in that's making its way through the digestive system. Again, nobody knows this. All these are speculations. So I can't say definitively yes or no. But the numbers would argue that something facilitated that disease happening in people in England.
Does it have to be ingested, or is there another way?
We don't know. So does it have to be ingested? We just don't know. I mean, it's not a very infectious disease. If you know somebody who has Creutzfeldt-Jakob, it's not like you're going to get chickenpox because you told Grandpa goodbye or something like that. But at the same time, we just don't know how it's transmitted. It's not a widely transmitted disease, which is good. But beyond that, we just don't know. Yeah. There was, and getting back to the eating other organisms, there's a disease in humans called Kuru that supposedly arose from tribes in, I believe, Southeast Asia that were known to be cannibals. And so it's, but again, that's anybody's speculation. Don't know. I will say this about the prions. As I said, prions are extraordinarily stable. You can't break them down, or you can't denature them by baking. It takes 700 degrees before they will completely come apart. So that might make you think that maybe they could make it through digestion or something like that. But again, yeah.
To clarify on that, prions are the ones that if you get one misfolded, then like they cause others to be misfolded?
Right. Prions are the proteins that, when they misfold, they favor the misfolding of other copies of themselves. Has to be the same proteins, not other proteins. And we're seeing these misfolding diseases now are popping up in a lot of ways. Alzheimer's, Parkinson's. There's a variety of diseases now that are thought to arise from misfolding of proteins. Not necessarily as prion diseases, but as misfolded proteins.
So what is the difference between just a misfolded protein?
Yeah. So the prion disease is one that's fairly active, and you can see that the protein causing the misfolding of additional proteins. In the case of Parkinson's and things like that, I don't believe that it's thought to be a causing a misfolding that's happened, but rather arising from misfolding. The misfolded proteins aggregate. That's the problem. And so these are all in nerve tissue, and so that's why all of these, certainly all the prion diseases, result in dementia, because the brain, and they don't last very, I mean, this is where the infectious, not the infectious, but the aggressive part of it occurs. Once you get diagnosed with Creutzfeldt-Jakob, for example, you probably have a year, and maybe half of that sane. You know, it doesn't last. You don't last very long with that.
How do you reverse non-competitive inhibition?
How do you reverse non-competitive inhibition? Take away the inhibitor. Because you can't outcompete it, right? You can't outcompete it. And so taking away the inhibitor is really the only thing that you've got to do that. Add more enzyme. So the question is, does the inhibitor let go of the enzyme? And the answer is certainly. These are not covalent interactions. But since there's plenty of other enzyme for it to bind to, once it lets go of one, as long as it's chemically stable, you'll have a fixed percentage of enzyme inhibited when you've got it present in a mixture.
Any questions?
Yeah, no, okay.
Yes, sir.
Calculating the overall part.
Okay.
When in recitation, I know you said that you're going to use all the pKa values.
Mhm.
Are you going to give us the pKa values from that kind of in the chart that you did in recitation?
I haven't decided. I will always give you enough information to solve the problem. That I'm not going to ask you to read my mind. You wouldn't want to. Yes?
And then kind of going on to that, we don't have the pKa's memorized, but we do have the equations that use pKa's memorized.
I give you every equation you need.
Okay.
Yeah. So Henderson-Hasselbalch, you'll have that. Any equation that you need on the exam. No memorizing of equations. Nope. I want you to know how to use the equations. That's what I want. Clear as mud? All right, guys. Well, it looks like we have exhausted the supply of questions. I will see you tomorrow. I wish you the best on the exam on Wednesday.